Hello, and welcome to Session 2 of Ivy Tech's Psychology 101, Introduction to Psych. Hello, and welcome to Ivy Tech Community College's Psychology 101, Introduction to Psychology, Session number 2, where we'll be discussing neuropsychology and the biology of the brain. This is Part 1, the nervous system. The nervous system of the human is consisted of two basic parts, the central nervous system, the CNS, and the peripheral nervous system, the PNS. The central nervous system is comprised of two basic parts, the brain and the spinal cord. The spinal cord is a collection of neurons and supportive tissues running down the base of the back, down the center. It's protected by the bones of the spinal column. Your brain is an approximately 3-pound collection of nerves and neurons. If you take your two fists and place them together, that is the approximate size of your brain. The nervous system has two basic parts, the central nervous system and the peripheral nervous system. As we've already stated, the central nervous system is comprised of the spinal cord and the brain. The peripheral nervous system is comprised of the somatic nervous system and the autonomic nervous system. A simple way to remember these is if you've ever studied Latin or know something about prefixes, soma loosely translates to the word body. And most of us understand that auto is very similar to the word automatic. So the somatic nervous system covers your major muscle movements, while the autonomic nervous system controls those things that are automatic that you can influence but you can't control. The autonomic nervous system is comprised of the sympathetic and the parasympathetic systems. The division of the nervous system that consists of the brain and the spinal cords are again the central nervous system. The brain is the central feature. Your decisions, your actions, your emotions are primarily based in the brain. The spinal cord communicates with the peripheral nervous system. So this is another layout of the nervous system with some of the information about what each part does. The central nervous system processes, interprets, and stores information and issues the orders to the muscles, glands, and organs, while the peripheral nervous system transmits information to and from the CNS. The somatic nervous system controls the skeletal muscles, so everything that you're doing, whether you're writing things down or you're sitting up straight or you're walking, is controlled primarily by the somatic nervous system. Everything else that you really can't control but you can influence is governed by the autonomic system. You can't consciously control your heart, but you can influence your heart rate by thinking about something that's exciting or scary or being around somebody that you have an emotional attachment to. Those influences to the autonomic system are impacted by the brain and the spinal cord and somewhat the somatic nervous system, but you truly can't control the autonomic system. If you want to test this, simply try and think about stopping your heart. Hopefully it didn't work, because if it did, that's a pretty serious problem. What we know is we can influence these things, but we can't control them. With the PNS, the division of the nervous system is between the autonomic and the somatic. The somatic nervous system allows us to walk, run, sit upright, write on paper, type, pretty much anything that we voluntarily do. It also carries muscles to and from the sense organs, like your skin or your tongue. The information that we gather from this is processed by the central nervous system. The autonomic nervous system is self-governing, and it's comprised of the sympathetic and the parasympathetic systems. What the autonomic system controls are things like your heart rate, sweating, anxiety, eye dilation, and digestion. The subdivision of the autonomic system that is responsible for mobilizing the body in times of stress is the sympathetic nervous system. It is the emergency system, and we call this the fight or flight part of the system. If you are presented with a stimulus that is frightening, scary, or startling, your sympathetic nervous system automatically responds. And so the fight or flight system comes alive. There is some theory to say that it is fight, flight, or freeze, but essentially freezing is just a variant of flight. It is the decision of the sympathetic nervous system and the central nervous system that running or getting away is impossible or improbable, and so the person just freezes and doesn't do anything. It's a hyperactive point in the sympathetic nervous system. And so the sympathetic nervous system is controlling dilation of pupils, increased sweating, increased heart rate, and it secretes adrenaline. So again, imagine you're walking down the street and it's dark and something jumps out at you. What your sympathetic nervous system is going to do is it's going to increase sweating because your body is going to start to heat up with all the activity of fight or flight, and the sweating will cool you down. Increasing your heart rate will get more blood throughout your system, will get more energy out there because it will transmit reparative cells. It'll start the glucose going through your body. And adrenaline is that component that allows you to be hypervigilant or use large amounts of energy very quickly. The dilation of the pupils is very interesting because it allows for you to take in more information. And so usually your pupils dilate due to stimulus of light, but here your pupils will appear to open up and take in more information. The parasympathetic nervous system is what gets you back down to a resting and balanced state, or what we call homeostasis. This, after the sympathetic nervous system has been activated, eventually you're going to get calmed down to the point where you need to get your heart rate, your breathing, and your digestion to calm down. One thing that happens that we didn't mention previously is that during a sympathetic nervous system activation, digestion tends to shut down because your body realizes that the food and the calories that are inside of you, it might be more beneficial to have those at the ready so that if you need a longer duration of fight or flight, you will have that opportunity. However, when the parasympathetic nervous system kicks in, digestion also kicks in. And so the parasympathetic system will constrict the pupils, slow down the heart, constrict the bronchi in the lungs, and constricts the bladder so that you can secrete urine and you can use the restroom and get everything back to homeostasis. What we're going to do now is look at the study of the brain. And the brain is one of those very interesting things that we can't study directly in many cases. One way that we can study the brain, though, is through case study. Case studies is a research method in which the brain can be looked at after the person passes away, and if we know what type of injury or disease they had, we can look at things backwards and say, if this individual had a traumatic brain injury and part of the frontal lobe was compromised, and we also know that at the same time their personality changed significantly, then we can make a correlation that the frontal lobe must have something to do with personality. The case study that we're going to talk about in Psych 101 that really illuminates this whole point is the case of Phineas Gage, which we'll talk about in a little bit. We can also look at patients who have had strokes and have had specific areas of their brain damaged. For example, if somebody has a stroke and we can find out that it was located in the temporal parietal area that has the Broca's or Wernicke's area, and they also have a correlated deficit in the way that they either speak or interpret speech, then we know that those two little areas have something to do probably with speech or language. We can also do experimental interventions. The lesion method is a method in which we can damage or remove a section of the brain in animals and observe the effects. Obviously, this is not something that most people volunteer for. However, the lesion method is loosely what was used in research when, in the early 1900s, doctors were performing frontal lobe lobotomies. These were not lesions, these were severing the frontal cortex, but they also could see in humans, if they severed the frontal cortex, what type of behavioral and neurological deficiencies showed up. And so in animals, you can go in and actually do lesions or electronic stimulation, which we'll talk about in a little bit, that once that lesion is in there and that part of the brain is compromised, we can see what impact it has on behavior. A very popular lesion method study that was performed in the history of psychology was a study on rats to see what part of the brain actually houses memories. So what they did is they taught mice and rats to run a maze, and then they would put lesions in parts of the brain that they believed that would house the memories. What they found is that memories are not housed in any one part of the brain, that they did lesions on almost every single part of the brain that they could think of, and the rats and mice still remembered how to run the maze appropriately. So the information that was gathered from that was that memories are not housed in one part of the brain that we can go in and pull out. Rather, they're housed in a big neural network, which is very similar to the internet, that most information is not housed in one place, it's housed in many places, and we can access it from different locations. There's also stereotaxic surgery, where we can implement electrodes into the brain and activate parts of the brain to see what happens. So what we're seeing here is the case of Phineas Gage. Before I show you the video, I want to explain what you're going to see. Phineas Gage was an individual who was working in the late 1800s on putting in the Transcontinental Railroad in the United States. Part of his job was helping to set iron rods on the ground where there was gunpowder that would allow the ground around the iron rod to fall in on the iron rod. So the hole was dug, the post was put in, about a 3-foot iron rod that was about 1.5 inches in diameter. And once the gunpowder was lit, the ground around the iron rod would fall in, and they could then move along and put more iron rods down. One day, through no fault of Phineas, as the story goes, they lit the gunpowder a little too soon, and Phineas was standing above the iron rod. The iron rod shot up and through his chin, taking out one of his eyes and a large portion of his frontal lobe. So what we'll be able to see here is Phineas, and this is not really Phineas, but this is a representation of what Phineas actually occurred, or what happened with Phineas. We'll see the iron rod go through the left eye and out the top of the head, taking out a large part of his frontal lobe. What's interesting about this story is that Phineas did not die. Quite the contrary, Phineas was perfectly fine except for the fact that he lost vision in his left eye because his left eye was severely compromised by the injury. What happened to Phineas is that he went from a very calm, caring, good worker, good family man, and as reports go, he turned into somebody who could not hold a job, could not be a part of his family anymore because his behavior and his personality changed so significantly. So looking at this through a post-mortem lesion study, because the lesion wasn't put in intentionally, and partially a case study, we could see that damage to the frontal lobe impacted Phineas's personality. Therefore, we can correlate and say that the personality is probably housed inside of that frontal lobe. So a tragedy to a gentleman actually became a very important contribution to neurological sciences. We also can do electronic or electric brain recordings, where we can use things like EEGs to detect electrical activity in the brain. EEGs are brainwave recordings that are interpreted by computers, and different brain waves for different states of consciousness. So we can see alpha waves and beta waves. We can see that when people fall asleep and they're in deep sleep, they have theta waves. We can also help to identify tumors in epilepsy because there are multiple receptor points on the skull, and if somebody happens to have a seizure while they're connected, that seizure will normally start in a very centralized location and sometimes spider web out. So EEGs are very important for this type of research. We also can do brain imaging. One of the more popular and older versions is positron emission tomography, or PET scans, PET scans. It records the metabolic activity in the brain as they occur. So basically it's looking at energy consumption, and the correlation there is that as the parts of the brain consume more energy, theoretically it must be working more. So if the frontal lobe, when you're thinking about something, is consuming more energy, we believe that that means that that part of the brain is actually doing work. So it uses a radioactive glucose-like substance as a tracer and allows us to study which parts of the brain are active when performing mental tasks. And we get a very colorful picture from this, the reds and yellows showing activity, the blues showing lack of activity. So if we look at this picture, we can see that both eyes are open on the upper right picture, and the ears are closed. So you can see a very large part of the brain, the frontal lobe, which is the top part, is activated, but also the occipital lobe is very activated. You can see it's very red right there. Eyes open and ears open, the upper left-hand picture, you can see that the Broca's and Wernicke's area are being activated, and those are in the parietal area of the brain, the sides, and the eyes are open, so the occipital lobe is actually very, very activated there. We can also look down here, and we can see with the eyes closed and the ears open that the occipital lobe is not activated hardly at all. It's slightly activated because this person is probably having some visual content that their occipital lobe is making up or remembering, but you can see a lot more activity over here when the ears are open. And when the eyes are closed and the ears are closed, really we don't have a lot of auditory stimulation going on, but the occipital lobe back here starts to reactivate. So what's really interesting is when we tell children eyes open, ears open, the most brain activity is actually occurring at that point. We also have CT or CAT scans, which are X-rays interpreted by a computer. We don't totally use this one in research. This is more of a practical type of brain imaging device, and it can reveal the effects of strokes, injuries, and tumors. Finally, probably one of the most interesting ways that we can do brain research is by using MRIs or fMRIs. MRIs uses radio waves and strong magnetic fields to give us a picture of the brain, and it can actually do slices. By controlling the depth of the MRI machine, it can give us images of the brain as if it was transparent, so we can go deeper or more surface area. And with the new fMRI machines, the functional magnetic resonance imaging, we can actually see the functioning level of the brain. We can see it in action, which is very interesting for researchers. We're going to go ahead and look at the parts of the brain on a very simple basis. We're not going to go deep into all the components of the brain, but just understand that the brain is a very complex structure, and we're going to go over some of the very basic parts of it. The hindbrain is the oldest part of our brain. Its major structures are the medulla, the pons, and the cerebellum. These are the most functionally important parts of the brain. That's what keeps us alive. The frontal part of the brain, the frontal lobe, is actually what makes us human, but this is the part that keeps us alive and keeps us aware, and it keeps us engaged with our surroundings. So we look at the EEG of a normal brain, and a brain that has all of its components intact, and we can see that there's going to be these different brain waves. Again, we've already talked about this before, but we can see these beta waves on the aroused section. In the relaxed section, we can see that we're going to have what are alpha waves. These are considered to be alpha waves. And these are going to be theta waves down here in deep sleep. So When we are aroused, we have these beta waves. When we're relaxed and starting to fall asleep, we have alpha waves. Down here, we have theta waves, where the brain is not active, but when you're asleep, your brain is still going. It does not shut down at any point. In the hindbrain, we have a lot of different components. First one is pons. The pons are important for sleep and arousal because they act as a bridge between the medulla and other areas of the brain. The medulla is the structure of the hindbrain that regulates the autonomic responses like breathing, swallowing, and blood circulation. Obviously, the medulla is very important because it allows us to sleep and it allows us to think, because if we constantly had to be thinking about breathing and keeping our heart going, we wouldn't have time to do anything else. So the medulla is, it regulates the autonomic responses, and it's incredibly important for our survival. The cerebellum is the part of the hindbrain that coordinates muscle movements. So your cerebellum is constantly regulating your posture, and it stores muscle memories, those memories that are related to skills. So once you've practiced something over and over and over, and you can do it without thinking, such as driving or writing, it's your cerebellum that's actually controlling those muscle movements. And an interesting thing to do is, if the next time you're walking upstairs or the next time you're walking down the hallway, actively think about how you're walking, how your legs are moving, how your arms are moving, what you're looking at. Because when your frontal lobe tries to do something that your cerebellum normally does, you're normally going to act very awkwardly. This actually has some practical application in detecting deception. If you want to know if somebody is trying to act normal, then oftentimes their frontal lobe is trying to get them to act normally when their cerebellum knows exactly how to act normal. If anybody who's listening to this is a fan of poker or plays poker themselves, they'll notice that if somebody is trying to lie to them, or what poker players call bluffing, their frontal lobe will say, I need to act normal, and they'll start to actively act abnormally. Sometimes they'll drop their poker chips or play with their hands in a weird way because they're trying to act too normal. That's their frontal lobe trying to do something their cerebellum is good at doing. The midbrain is a major division of the brain that contains fibers known as the reticular formation. It's sometimes considered part of the hindbrain, and sometimes it's considered to be apart from the hindbrain. So it just depends on how we look at it. The reticular information are the nerve fibers passing through the midbrain that control things in the autonomic system, like arousal, attention, and reflexes that are involved in breathing and sneezing and coughing and the vomit, the gag reflex. The brain stem is composed of these components. It's the reticular formation, the medulla, the pons, and the cerebellum. This is the most basic part of your brain, but by far very important. If we look at most animals, they have the same basic setup, especially if we look at the comparison between early mammals and humans. The brain stem is very similar, but as we get closer to the newer parts of the brain, which are the outside parts of the brain, there's a significant difference. The forebrain consists of subcortical structures and the cerebral cortex, the cerebrum, the limbic system, and the corpus callosum. The corpus callosum is a wide band of neural fibers that connects the two hemispheres of the brain. So again, if you take your two fists and you put them together, the corpus callosum would be where your knuckles are touching each other. And it is the part that allows for left brain, right brain synthesis. The cerebral cortex, or the cerebrum, is the wrinkled outer layer of the brain. And this is what controls our voluntary movements, our emotions, thinking, consciousness, remembering, and learning. If you were to be able to take your cerebrum or cerebral cortex and remove it from your brain and put it out on a desk and lay it completely flat, it would be the size of 4 pieces of paper. So you take 4 pieces of paper, that 8.5 by 11 pieces of paper, and you put them out in a big square, and you can see how big the cerebrum actually is. So the question is, why is it wrinkled? It's wrinkled because we have to smush it inside your skull. It's a lot of area squished in, and so the more wrinkles, the more ability there is to have differing neural connections. If you take a look at the cerebrum or cerebral cortex of primates, their cerebrum is only about the size of one piece of paper, and so it's a much smaller part of the brain for them compared to us, and that is one of the reasons why if you look at their brains, it's actually a little bit smoother because it doesn't need as many wrinkles to fit inside of the brain. The limbic system is a system of interconnected subcortical structures that regulate a variety of emotions and motivated behaviors, such as hunger, thirst, aggression, and sexual behavior. The hypothalamus is the part of the brain that actually controls your thirst and your temperature regulation. And the amygdala, the hippocampus, and other parts of the limbic system have other types of simple controls. The amygdala is the part of the brain that actually controls anxiety, fear, and fear reaction. So it's a very big part of the autonomic system. The thalamus is a subcortical structure that relays information, such as sensory information, to the cerebral cortex and other parts of the brain. So the thalamus is basically like a big switching post. It's where if you can visualize the old style operators where they have all the plugs that they have to do to connect phone calls, it's kind of what the thalamus does. Information comes into the thalamus and it sends it out to other parts of the brain. We'll talk about neurons in another section, so I'm not going to go into that right now. But the cerebral cortex is, again, this wrinkled part of the brain. And what we're going to see here is the actual 4 lobes of the brain. The visual cortex is housed primarily in the occipital lobe. So if we were looking at this as a human, this part would be the eyes. So your eyes would be about right here. So the frontal lobe is right above your eyes, and here would be your nose and your nasal cavity. So the frontal lobe is the front part, and it is a major part of the brain for humans. It houses a lot of the motor cortex, which we'll talk about later. But the motor cortex is actually, the motor sensory cortex is right next to it. These two are very, very interesting because the somatosensory cortex is actually a layout of your body. And it's not sequential. It's not like the feet are down here and then your shins and then your knees and then your hips and then your torso and then your head. But it's in a very odd order. But anytime you actually sense something, if you sense something on your hand, it's going to come in right here into the hand part of the somatosensory cortex. What's interesting about this is if you lose feeling in a part of the body, let's say that you have to have your feet or a leg amputated, and the leg section is right here, you will actually have the somatosensory cortex will still have that part of the body, and it will still associate it in your brain with the part of the body that's now missing, but the places in the somatosensory cortex that are right next to that missing leg will actually start to encroach in on the leg portion. So your brain will believe that your leg is still there, but when you touch, say, your cheek, and the cheek has encroached in on that leg portion, your brain will interpret it as your leg actually feeling something. And that's how brilliant and how kind of silly your brain actually is. It believes the leg is still there, but because the leg is no longer there and no longer sending in information, another part of the body actually encroaches on that cortex, and so when you touch that part of the body that still exists, your brain will interpret it as the part that's missing. We have down here the temporal lobe, and this is where we get the auditory cortex and the Wernicke's area. The Broca's area is actually in the frontal lobe, but its borders on the temporal lobe. Wernicke's area and Broca's area are where we get most of our receptive and expressive communication skills. So the parietal lobe has a lot of our association cortexes, or somatosensory cortex, and the frontal lobe is the association cortex. This is where we get most of our higher order thinking and what makes us truly humans. And so again, we see our picture here of Phineas Gage. Obviously not really Phineas Gage, but it's an interpretation of what happened to him. The frontal lobe consists of the primary motor cortex, which controls voluntary muscle movement. So if you are thinking about, I need to move my arms in a certain way because I'm learning a dance, your frontal lobe is actually doing it until you've gotten into your muscle memory, and then it transposes it to another part of the brain. The frontal lobe is responsible for decision-making, goal-directed behavior, and executive functions, those higher-order thoughts that you tend to have. The parietal lobe, or sensation of touch and bodily position, it also has that primary sensory cortex. It processes the body sensations. Temporal lobe, again, the Broca's area borders on the frontal lobe and the temporal lobe, but the temporal lobe processes information concerning hearing and smell, equilibrium, because we do know that the equilibrium is controlled primarily by the temporal lobe, but also in association with the auditory portion of our sensation and perception system, our ears. The left temporal lobe is the language center. So when you're hearing things and trying to come up with words, it's the left temporal lobe that's actually controlling your language. The occipital lobe processes information from the eyes. So the sensations coming into our eyes are transmitted through optic nerves to our occipital lobes, and it processes that information. And so the sensation of vision turns into the perception of what we are seeing. We look at the difference between the left and the right hemisphere. The left hemisphere is the interpreter for right-hand touch and movement, speech, language, writing, calculation, sense of time, rhythm, and complex movements. While the right hemisphere controls the left-hand touch and movement. The right hemisphere also is big on facial recognition, nonverbal imagery, so visualization. It has the visual cortex, perceptual skills, recognition of and expression of emotion. We know that we know this primarily from looking at case studies of people who have had strokes or consistent seizures. We know that the right hemisphere controls these parts and the left hemisphere controls other parts. The cerebellum, or the cerebral cortex, regulates movement and balance and plays a part in analyzing sensory information, solving problems, and understanding words. So those things, again, that make us very human, that's what's happening with the cerebellum. The thalamus, we've already talked about a little bit, but it relays information. So it's located in the center of the brain, and it includes all sensory messages except for the olfactory bulbs. So vision goes through the thalamus, taste goes through the thalamus, touch goes through the thalamus, hearing goes through the thalamus. Olfactory bulbs, the smell does not go through the thalamus. The hypothalamus and pituitary gland. Pituitary gland is probably something you may have heard of in biology or maybe just heard of in conversation. It is a small endocrine gland that releases and regulates other endocrine glands. Pituitary gland is huge for the growth, especially during early childhood and puberty. Pituitary gland, if you have an issue with your pituitary gland, sometimes it will cause you to under or over grow. And so when you see people who are incredibly tall or incredibly large, sometimes there's an issue with the pituitary gland. The amygdala, again, is responsible for those autonomic responses such as arousal, emotional regulation, anxiety, depression, and emotional memory. The hippocampus, hippocampus meaning seahorse, and because it looks like a small seahorse, is responsible for the storage of new information. If there's a deficiency in the hippocampus, people tend to have trouble storing new memories, and sometimes we'll have a disorder called anterograde amnesia, which means they can't store new information. It enables us to form spatial memories, and it also helps us compare sensory information with what the brain expects about the world. So your hippocampus is incredibly important for going into new situations. Your brain prefers to stay in situations that it knows and it understands because it can use mental shortcuts. But the hippocampus allows us to take what we know and compare it with new situations so we don't constantly have to do micro experiments to survive in the outside world. The corpus callosum, again, as we talked about, is the thin bundle, a very dense bundle of nerves that connect the left and right hemisphere. We know the corpus callosum is important, but it's not imperative that we have a corpus callosum to live. Some people will have what are called split brain surgeries. And so if we separate the left and right hemisphere through a split brain surgery, sometimes people with epilepsy will be able to function much better because the hemispheres cannot communicate directly with one another. So a seizure might be happening in the right hemisphere, but the left hemisphere is still okay, and it allows them to still cope and allows them to still live their life with some quality. And so we look at split brain experiments. What happens if we separate the corpus callosum? Because information is being passed from the right eye to the left hemisphere and from the left eye to the right hemisphere, we can see that people can actually see two different things at the very same time. And if they're together, they'll actually create some kind of amalgam picture. So you look at A, look at the center of the slide, and so the person is with their right eye taking it into their left brain and their left eye taking it into their right eye, and then they ask people to point to who they saw. They will actually create an amalgam person. It's because the left and right hemisphere is actually trying to come together at some point. We talked in session 1 about a gentleman by the name of Kim Peek. Kim Peek was born with agenesis of the corpus callosum, which gave him some incredible skills, like being able to read the left and right page of a textbook simultaneously, and he could retain information at about a 98% rate in some cases. This is exactly what Kim was born with. Instead of having a split brain experiment, he actually was born split brain. So if we look back at those pictures, we see that patients were presented with information on one or the other side of the brain. Patients identified verbally the picture to the right, the boy. When patients were asked to point to the face they had seen, the patients pointed to the left picture. Now, do we have different brains between the genders? Research has shown that after studying about 49 studies in the sex differences in brain anatomy, most researchers find, and this is a macro-analysis of a bunch of different studies, the researchers found small differences between the two groups and larger differences within the groups. There do appear to be sex differences in lateralization of language, to which side of the brain or both sides of the brain does language primarily occur. Males show left hemisphere activation only when we're talking about language, whereas females show left and right hemisphere activation. Females do tend to have more gray matter in their brain, gray matter being the neural makeup of the brain. And so what do these differences mean for behavior? The supposed differences are actually more in tune with stereotypes than anything else. Brain differences does not necessarily explain behavior or performance. This is one of those indications or one of those examples of where size really doesn't matter, because we look at history and we see that people like Albert Einstein had a brain that was fairly significantly smaller than the average brain for a person his size. It was between 10 and 15% smaller than the average size brain. And I would say that Einstein had used that smaller size brain much to his advantage. Sex differences in the brain could be the result rather than the cause of behavioral differences. So instead of the fact that we know that there's lateralization with females more often than males, it could be the result of these issues, and rather than the actual cause. And that's going to bring us to the end of our first section of session 2, neuropsychology. We're also going to be discussing neurons and neural pathways in the brain, and also the role of neurotransmitters in our next lecture. Hello, and welcome to Psychology 101, Introduction to Psychology for Ivy Tech Community College. My name is Professor Brad Mitchell, and this is Session 2, Part 2, Neurological Implications of the Brain, and we're going to specifically be talking in Part 2 about the neuron and neurology. So we'll start out by looking at neurons, the types, components, the way they communicate, and also the role of neurotransmitters. There are two types of neurons. There are afferent and efferent neurons. Afferent are sensory neurons, or nerves that carry information from the receptor site to the spinal cord and then to the brain. Afferent neurons detect information from the environment and pass information through connector neurons, which activates motor neurons. Efferent or motor neurons are nerves that carry information from the spinal cord and the brain to the receptors. While these two terms are very similar, afferent and efferent, one easy way to remember this is that you must sense something before you can act on it. So you must sense that there's a change in the environment or something touching you before you can move. For example, if you put your hand under some water and it's hot, the afferent neurons, the sensory neurons, are going to send information to your spinal cord in your brain, and the efferent motor neurons are going to have you move your hand. So A comes before E, and sensory comes before motor. The components of a neuron. So we need to look at the actual major components of a neuron. We're going to start down here at the left-hand side, and these tentacles are called dendrites. Dendrites receive information from the outside world or receive information from the previous nerve. The information passes along this arm and goes into the soma or cell body. The inside purple part that I'm highlighting right here is the nucleus of the soma or cell body. Once the information is determined to be able to be passed along, the information goes down this arm or leg part of the nerve called the axon. And these tiny fatty globules that cover up the axon are called the myelin sheath. Once the information goes down through the axon, it's going to go through the axon terminals, down here, these arms coming off the axon, where neurotransmitters are held in the axon buttons. Information is then sent out through the axon terminals to the next set of dendrites on the next nerve. Once again, the dendrites are going to receive information from other neurons and transmit it forward to the cell body or the soma. The cell body or soma keep the neuron alive and determine whether it will fire. And the nucleus inside of that soma carries most of the information. The axon is the extending fiber that conducts impulses away from the cell body and sends it to other cells. Glial cells are a special type of cell found in the nervous system that are formed in the myelin sheath. And these glial cells remove waste and occupy space of dead neurons and provide structure. So most of your brain is actually consisting of these glial cells, this fatty tissue that we now believe also has a strong impact on our reflexive actions, those muscle memories that we don't have to think about. The information is primarily thought to be held inside the myelin sheath with these glial cells. How do neurons actually communicate with one another? The axon is going to carry the information from the soma or the cell body, and it's going to send a neural impulse. That neural impulse is going to send a neurotransmitter out of the end of the axon, or the axon terminal, and the neuron is actually going to go in between two nerves through what's called the synaptic cleft. The neurotransmitters that you see being thrown out of the axon and the axon terminal then go to other nerves to see if their neurotransmitters will fit with that receiving dendrite so that it can pass on the information from one nerve to the other. So we see that in that synaptic cleft, we have neurotransmitters that may or may not fit the lock of the receptor sites. It's much like a key. Those six-sided or eight-sided little Tetris wedges are the types of neurotransmitters that are being sent that actually will activate the postsynaptic membrane. Once those fit the lock, they're going to send information. You can see on the far right-hand side, there's some neurotransmitters that are going to be transmitted that aren't going to fit the lock, and therefore that information will not be passed on. But there are billions and billions of nerves in your brain, and so each nerve has 1,000 to 10,000 different dendrites and axon terminals, and so they can create billions upon trillions of different connections. So if one receptor site isn't effective, there's going to be others that will. So once that information is received, once it hits the threshold, it's actually going to send the information on. So receiving neuron averages the excitatory and inhibitory incoming messages to determine if it's reaching its firing threshold. What does it mean to be on the firing threshold? It means that whether or not the neurons will fire, the receiving neuron will fire, will send the information through the soma down the axon, is once it hits the threshold, it's going to fire, and if it doesn't reach the threshold, it won't fire. It's an all or none situation. The strength of the firing doesn't alter. Okay? A lot of times this is a confusing concept, so I want to talk about the all or nothing concept here for just a second. So it's considered an all or nothing firing, and so the analogies that I like to use is to think about either a handgun or a doorbell. It doesn't matter how hard you squeeze the trigger of a gun. You can squeeze it as hard as you want or as soft as you want, but you're going to get the same result. Once the threshold is passed, the bullet is going to fire. Same thing as a doorbell. If you go to your door and you punch the doorbell as hard as you can, or you barely touch it until it finally makes a connection, it doesn't change the strength of the actual bell that rings. Once the action potential has been hit, the impulse goes, and it's all or nothing. You can't lightly ring the doorbell. You can't lightly shoot a gun. Once you reach the threshold, it's going to fire. The same thing with your nerves. Once you have enough neurotransmitters activating the next nerve, it's going to fire. So we're going to talk about a few basic neurotransmitters. This is by no means a complete list of all the neurotransmitters, but this is a collection of 4 or 5 neurotransmitters that are incredibly important in the realm of psychology and psychiatry. The first one is acetylcholine, or ACH. This neurotransmitter's role is to link motor neurons and activate muscles. So a deficiency in acetylcholine will actually cause problems with movement. It plays a very strong role in memory, and therefore it can have an impact on Alzheimer's disease. It also has an impact on arousal and motivation. Probably the most interesting, in my opinion, and most dominant neurotransmitter when it comes to psychiatry is dopamine, with a very close second coming with serotonin. Dopamine is an inhibitor that is involved in the control of voluntary movements. And so if you think about the disease of Parkinson's disease, the two people that are usually most people know who have Parkinson's disease are the boxer Muhammad Ali and the actor Michael J. Fox. If you see them symptomatic with their Parkinson's disease, you will realize that they're having tremors, they're having trouble controlling their movements. This is because they have a deficiency in their dopamine, and their receptor sites are not good at receiving information from dopamine. So the dopamine, which is an inhibitor to voluntary movements, is not effectively helping them, and so they have a lot of involuntary movements that they can't control. What's interesting is if you have too little dopamine, you have Parkinson's disease. However, there's also a theory that too much dopamine can lead to schizophrenia. So people who are being treated for Parkinson's disease are given a drug called L-DOPA, which makes the dopamine more effectively used in the body. But if they're given too much L-DOPA, they'll start to come down with schizophrenia syndrome, meaning that they're going to have symptoms of schizophrenia, but they don't really have schizophrenia. The same things happen for people with schizophrenia. If they take too much antipsychotic medication, they'll start to have tremors because the dopamine levels are too low, and they will start to exhibit Parkinson's syndrome, meaning that they don't have Parkinson's disease, but they will still have the symptoms. Serotonin is another huge neurotransmitter when it comes to psychology and psychiatry. Serotonin affects emotion and mood, and it is probably the most important neurotransmitter for sleep, and when there is a dysregulation of serotonin, there is a strong indication of depression. This is why one of the earliest and probably one of the best prescribed medications for depression are called SSRIs, selective serotonin reuptake inhibitors. What happens is that serotonin is released into the synapse. Once it's released and it goes over and it activates the receiving neuron, it then goes through a process of reuptake where it's sucked back into the sending neurotransmitter, or the the neurotransmitter is sucked back in. Serotonin is sucked back in to the axon terminals and axon buttons of the sending neuron. Selective serotonin reuptake inhibitors go in and act as an antagonist to the sending neuron, and it doesn't allow the serotonin to go through the process of reuptake where the serotonin is sucked back into the sending neuron, and so the serotonin stays in the synapse longer and activates that receiving nerve multiple times, and it makes people believe, because of the neurology and because of the amount of serotonin left in the synapse, that they're actually feeling better. Endorphins. Endorphins are a morphine-like neurotransmitter that reduce pain by inhibiting nerves that transmit pain messages to the brain. What you're going to find as you study psychology is that everything happens in the brain. If I stub my toe, it sends an impulse to my brain, and pain is actually felt in the brain. And so if we have endorphins, or we have something like morphine that will act as an inhibitor to the neurons that pass information to the brain, then you won't feel the pain. The impulses are still going to be sent up, but they're going to stop at the brain and you won't perceive them. So the sensations stop, the impulses stop, and the brain does not perceive the actual pain. GABA is involved in excitability, arousal, and anxiety. And this is another neurotransmitter that has a very strong impact in the role of psychiatry or medication management with psychiatric medications. And so that takes us to the end of this small section, part 2 of session 2. I hope that you have gathered information that will help you understand how the brain interplays with the rest of the body, and how the central nervous system and the peripheral nervous system interact, and how neurotransmitters play an integral role in brain utilization and emotions and behavior.